present close together or whether they are present far away, whether any possibility of combinations or recombination happen between them, or what are all the possibilities of not having recombinations. All these factors decide how the genes are getting inherited, right? And when two genes are, the probability of two genes are very high for not getting separated, generations after generations, we call that probability as linkage, right? And when you have linkage, you have the option of identifying one locus when you know the other locus, the neighboring locus, right? If you know the one locus which is linked to another locus, if you identify one, you will automatically identify the other. That is the principle we use in linkage mapping, or what you call the otherwise known as the QTL mapping, if the trait is a quantitative trait, right? Okay, we will go further. Now we know that if we have a linkage, then it causes a disequilibrium. The disequilibrium of what? The disequilibrium of segregation ratios, or otherwise called the independent assortment. When you have the disequilibrium of independent assortment due to linkage, we call it as linkage disequilibrium, right? So this linkage disequilibrium is essential for mapping, whether it is linkage mapping or it is association mapping or whatever it is. When we take linkage for the mapping purpose, we call it as linkage mapping. If we take linkage plus other type of disequilibriums, we call it as association mapping or whatever it is, right? Now, going further, as I said, when you have linkage, there is an equilibrium, a disequilibrium. The disequilibrium changes the ratios, right? That means you will be able to calculate the frequency of the progenies which are in disequilibrium and which are in equilibrium, right? Which otherwise in linkage analysis we call as parental types and recombinants. The parental types are those which does not have any recombination, right? So they are the one with disequilibrium, and the types which are having equilibrium will be the recombinants. So if you calculate the proportion of that, we will be able to roughly estimate how proximal the two loci are. If the loci are far apart, you will have more number of recombinations, and if they are very close, you will have less number of recombinations. This percentage of recombination is taken as a measure of the distance between two loci, and it is measured, and that is known as, say, centimorgan, or otherwise it is actually called as morgan. And when it comes to 1%, 100% is 1 morgan, so 1% is 1 centimorgan. That is how the percentage or the distance between the two loci are calculated when the linkage mapping is done. How you do this? We know the classical genetics, we do linkage analysis. Linkage analysis can be done using 2-point analysis, 3-point analysis, or multi-point analysis. I am not going into those things which are very basic. However, now we have systems, we have computational systems to do this effectively, because doing a 2-point analysis is very easy. 3-point analysis slight more difficult, but if you want to go for multi-point analysis, it becomes very complicated. In such cases, you need to have a high computational power. For that, now we have the computers and softwares to do that, and we can whole genome wise, if you have several loci distributed across a genome, you will be able to do multi-point analysis using a high power computer, and you can come out with the sequence of the loci, how they are arranged on the genome, and you will be able to finally draw them in a map, and that is how the linkage map is drawn. Now for this, although you can identify the recombination fractions and the centimorgan distance between two loci at any point of time in your segregating populations, ideally an F2 population, then you will be, once you calculate the recombination frequencies between two loci anywhere in the genome, what is the maximum amount of recombination you can expect? You can expect 50%. Why 50%? Why 50%? Yeah. So you can have only 50% of the progenies or gametes that are getting distributed, right, in the progenies. So the maximum recombination fraction you can obtain is 50%. So the recombinations then therefore can take place, or quantity of recombinations can be in a range of 0 to 50%. Now if you say which are proximal to 50%, they are far apart. If they are the recombination fraction is 0%, you will say they are linked. That is how the estimates are made. Having done that, now your next objective is to find out what is the correct gene order, right? Because you have to place them on a map, right? To place them on a map, you have to compare subsequent recombination fractions starting from a point. So if you keep a point, the recombination fractions can go upwards as well as downwards if it is in the center, right? So comparing the recombination fractions up and down, you will be able to place the markers in a particular order, right? So what is then how will you say that which order is the best? How will you say that the best? I mean, you have ordered, say you made some 10 orders, and from that how will you say that which order is the best order? For example, A, B and C, I can put them B, A, C or B, C, A, whatever, right? In one cases I am getting 10 centimorgan between the flankings, okay, the extremes. In one cases I am getting 15 centimorgan. In one case I am getting 7 centimorgan. Which is the best order? If I arrange 3 loci in random orders, I am getting one case 15 centimorgan, the total length. In another case I am getting 10 centimorgan. In another case I am getting 7 centimorgan. Which is the best order? Which is the best order? Anyone? It is the one which gives the lowest. The order which gives the lowest length is the best order. Because if the orders are in exact positions, right, they will give you the minimum total length. This is called the criteria of optimality. Okay, I'll come to that. I'll come to that later. Okay, this is called the criteria of optimality. So when you identify the orders, you apply the criteria of optimality. There are several parameters for identifying the criteria of optimality, which I am not going to deal with, but understand that the lowest order is the order which gives you the lowest total length is the best order. Okay. Now, having done that, you have found out that when you make or when you order the loci, you can see that, for example, take A and B and C. If you add the distance between A and B and C, you will not get the distance between A and C. It will not be exactly equal. The reason is that the distances are not exactly additive. The reason for this is actually a phenomenon called interference. So when the first map of Drosophila X genome X chromosome was made by Sturtevant in 1913, in that paper he wrote like this. That time there was no evidence of interference. No one knows about what happens. But he wrote in the paper, the evidence so far as it goes indicated that the occurrence of one crossover makes the another one less likely to occur in the same gamete, which is exactly what is we have later on described as the interference. Why this interference occurs? There are several reasons for that, genetical reasons, chiasma suppressions and so on. If you go further into the recombinations, we can also see that the recombination over a chromosome is not, or the recombination frequencies over a chromosome is, or the chromosome landscape is not exactly similar in all places. In some places it has more recombinations, in some places the recombinations are less. When you have a point in the chromosome where you have frequent recombinations, we call it as a recombination hotspot. Whereas some places like centromere region, you have hardly any recombination happening. What happens is that this occurrence of recombination hotspots greatly influence your map distance estimation. For example, as it is shown in the picture here, although the physical length is this much, if the recombinations are very high, you have a tendency to overestimate the distance, and if the recombinations are very less, there is a tendency to underestimate the distance, okay, the distance or the recombination frequencies. Now, for this, there is no straightforward solution other than sequencing. If you sequence, you can get the physical length. If you look at the recombination fractions, there is no straightforward solution for this. Then what we have to do is, we have to have an adjustment. This adjustment is called, any idea? We call it as mapping functions. We call it as mapping functions. Mapping function is the mathematical adjustment of the observed recombination frequency to obtain the more realistic, more closer recombination frequency, estimated recombination frequency, so that you will be able to estimate the distance between two loci better. there are several of them. Several people have proposed several mapping functions. However, I mean, Morgan has proposed that there is no crossover at all. I mean, there is no interference at all. So there is no need of mapping functions. He says that there is no such interference at all. So he says that the distances are additive, perfectly additive. Where in case of Haldane and then it comes to Kosambi, the Haldane does not permit again interference, and Kosambi allows some sort of interference. In plant genome analysis, generally we use the Kosambi functions, whereas in case of human genome analysis, mostly they use Haldane functions. So I am not going to in detail, and this is the Kosambi's mapping function, right? These are all mathematical adjustment based on the pattern of recombination fractions over a distance observed, and then they have mathematically adjusted based on the statistical distributions. Now, having said that, now we know, we have a rough idea how to identify the distance between two loci, right? Then what are the different types of loci we are going to handle? That is very important. We can handle the loci, for example, at the position of a gene as a locus, or position of anything on the genome as a locus, right? Anything on the genome. May be gene, may not be gene, right? That means any fragment of the genome you can position on the genome, right, is referred to as a locus. And if that fraction is identifiable, then we call it as a marker. If that particular fraction is identifiable, then you call it as a marker. So if it is gene, we call it the gene locus. If it is of marker, we call it as marker locus, right? And as you know that the markers are also, they also follow the same Mendelian segregation, because the principles are same. Whether it is gene or a marker, the independent assortment, linkage, all these principles are, they hold good for them. Now, our idea is to map, right? First of all, what we have is, we don't have gene at our hand. We have the genome at our hand, right? How will you have the genome? You extract them in the test tube as the DNA, That is how, that is your starting material. Your starting material is in a small tube, you have an extract of the DNA pellet, okay? And that you are going to use for as a starting point of your mapping process. You know how to extract DNA, you know how to purify them, fine. The next point is, you have to identify what exists on this genome, right? For that, what you require is a set of small fragments of the DNA, which you can manipulate in a polymerase chain reaction, or you use some methods to break down the DNA so that they become small fragments, so that you will be able to analyze them or visualize them, right? When you break them, you call it as we call it as digestion, okay? So when you digest the DNA into small fragments and then you visualize those fragments, then you get the digestion based markers, right? Or when you use small fragments of the DNA as a primer to multiply a particular target genomic sequence, we call them as PCR based or polymerase chain based reaction based markers, right? We amplify them, or we can call them as amplification based markers. So this is how you are now identifying the genome. Okay, you are identifying the genome as small fragments over the genome, by some method. Now next step is you are going to visualize them. You are going to visualize them based on what? Based on their length. Based on their length. Okay. If the DNA length varies, their weight varies. When the weight varies, they move in a differential fashion in your gel, right? Then you identify them as different bands and you call them as different fragments. So when you have fragments and you have variation, we call them as fragment length polymorphism. Okay, fragment length polymorphism. So based on the fragment length polymorphism, whether it is PCR based or digestion based, you are identifying the genomic sequences or regions distributed across over the genome or the genome landscape, right? Now you have two parents, okay, two individuals, which are phenotypically diverse. Because if you have the same type of same type of individual, you will not be able to identify difference between them, right? If you wanted to identify the difference between them genetically, you have to have two individuals which are genetically different. So once you have two individuals genetically different, now you will be able to digest the DNA or amplify the DNA of the two individuals separately, and you will be able to identify which are all the fragments vary between the individuals, right? Because you already estimated the length of the DNA fragments. Now those fragments which are showing variations are called polymorphic fragments, and which are not showing are called monomorphic fragments. And these polymorphic fragments are our key to mapping. Okay, without polymorphism you will not be able to map. Okay, this is where the marker stands an important tool to mapping, right? Now you may ask why we use markers. We have already a lot of phenotypic variations. Why can't we use them for mapping, right? We can use them for mapping, you are right. But what is the problem? They are very less in number, number one. They are very less in number. If you take two individuals, how many variations you can count? Very few, right? It's countable. But you know that the genome is so large, it is so extensive, and having identified few traits or few countable traits, you will not be able to identify the exact difference between the two or exact genetic difference between the two organisms. And moreover that these traits are highly influenced by environments or their expression states, so many things. Because of that, the DNA fragments are more reliable for mapping than using any phenotypic traits or any expression based character like proteins, isozymes and things like that. And they are abundant, they are distributed all across the genome, and they are expressed, non-expressed, whatever it is, right? So you will be able to do you will be able to draw an exact picture of the genome when you use a genome-wide set of markers, because as I said, they are ubiquitous and abundant. There are several ways to identify them. I just given a list of them here. And having all these things, next stage is to our map construction. So as I said, we have taken two individuals who are genetically different. The next step is to make a hybridization between them. generate a progeny, an F1 progeny, and allow it to segregate. Allow it to segregate. When you allow the F1 progeny to segregate, what happens is that the one chromosome came from the parent 1 and the other chromosome came from, which came from the parent 2, they will combine and recombine and they will segregate into the whole population, right? Which means exactly what we did is we have digested the DNA. In fact, we have digested the DNA into a population, right? The whole population, the DNA from the parent 1 and parent 2 are naturally digested. They are combined and recombined. Now, you have an assortment of genes from the parent 1 and parent 2 in the progenies. Now, you know already which are all the markers which are polymorphic between parent 1 and parent 2. Now, will you be able to identify the same thing in the progenies? Definitely, you will be able to identify them, right? So, if you take any particular locus, you will be able to now segregate the progenies as parent 1 type and parent 2 type, right? You will be able to do that. Since you know we have the all the locus, every locus follows the Mendelian segregation. They also will show the Mendelian segregation. In such cases, you will be able to identify for any locus you take, you will be able to identify any polymorphic locus you take, you will be able to identify the parent 1 type, parent 2 type and the heterozygotes, where the both fragments exist together. Now, for this, you need to have a marker called codominant marker, otherwise it is not possible. So, markers have two different types of expressions. Either they can be dominant or they can be codominant. What do you mean by dominant? It is observed only in one parent. In other parent, it remains absent, or it is otherwise called null. The allele is not seen. We call it as null, a null allele. Okay? Such cases, we call them as a dominant marker. And when you have two different alleles coming out of the same locus, we call them as a codominant marker, which will exist as two different bands if the parent 1 chromosome and the parent 2 chromosome exist together, right, in the progeny. Okay? Now, the codominant markers are highly useful for mapping, because if it's a dominant marker, you will not be able to distinguish between homozygotes and heterozygotes, right? Okay. Now, ultimately what happens is that, as I said, by arranging all the chromosome, all the markers, polymorphic markers segregating across the progenies, you get a linkage map. So, I said okay, before that I will tell you, I said about the criteria of optimality, and next step is you have to go for the search algorithms which help you to identify what is the best order of the genomes. There are several algorithms we use, exhaustive search, heuristic search. We will see in the afternoon the practical, some use of some of these. Then finally, you check that the map drawn is okay by statistical significances, and you will be able to tell the map is okay. Okay, finally what you get is this. Now, having done that, our next step is to identify map the gene. Okay, that is what our exercise is doing, for doing that only, to identify or map the gene. To map the gene, what you have to have is a population. A population and the parents which are varying for your target trait, right? Varying for your target trait. Will you be able to, for example, if you wanted to map a gene for height, when your parents are having same height, will you be able to map them? You will not be able to. They may have several other differences, but your interest is to map the gene for height. But unfortunately, the plants are having the same height. okay, will you be able to map? You will not be able to map. So, it is very important that your parent should have the variation for the target trait, the trait of your interest. If you use a population without variation for your target traits, your mapping fails. So, that is the first thing you must keep in mind. Okay? Then once you have that, you have several options, several options to make. Okay? You can have doubled haploids, you can have F2s, you can have backdraws in bloodlines, you can have RILs, and so on, right? Yesterday, I think you have been discussing about the F2, whether F2 can be used for mapping. F2 can be used for mapping only under specialized conditions, if it can be replicated, right? Replicated means if they can be cloned, then F2 can be used for mapping. If they cannot be cloned, then it cannot be used for mapping, right? That is how, I mean, when you make the mapping populations, you must understand that the mapping population has a genetic framework bracketed between your two parents, right? Right? The parent 1 and parent 2. It cannot have any other genetic variation unless otherwise there is a contamination from outside. Your mapping population, that means if you pull across all the DNA of your mapping population and if you check them, you will get ultimately you get the same pool of your parent 1 and parent 2, nothing else, there won't be any difference, right? That is how that is why the mapping populations are important. Once that is done, as I said, you will be able to identify the loci which are segregating between parents. Now there is a confusion here. You have your traits, you know how the traits are segregating between the population, okay, you can do that, and you have several polymorphic markers segregating between the populations there are several, you have one trait segregating between the progenies and you have several markers segregating between the progenies. Then what I want is I wanted to identify the marker or the gene which is segregating for the progenies or for the trait. How will you identify? How will you identify? Bulk segregant analysis? You have several methods. You have said. One, somebody said bulk segregant analysis, somebody said correlation, okay, somebody said maybe analysis of variance, somebody said regression. It's all there. We have to use the crux is this. The markers, we don't know the phenotype. As far as markers are concerned, we don't know the phenotype. And what we know the phenotype, we don't know the genotype. That is the issue. Now we have to bring this data together. To bring this data together, we have to borrow the help of statistics. Okay? We have to borrow the help of statistics. So that is what we are doing. We are doing statistical analysis of the data so that we are identifying the probability of one marker associated to the trait better than the others. The one which is more likely to be associated with the trait better than the others. This is what is called the maximum likelihood method. Right? In statistics, we call it as maximum likelihood method. Identification of the best likelihood. Right? Here, we use two conditions, or otherwise we call assumptions. The probability of not identifying the marker or the association, or probability of identifying the best association, right? Then if you take the ratio of it, our assumption is that the marker is not associated to the or linked to the trait. That is the null hypothesis. Okay, null hypothesis. The alternate hypothesis is the marker is associated to the trait. Now, you take the ratio between the likelihood of having association to likelihood of not having association, then we call it as, the ratio is called the likelihood of odds, or otherwise called the log of odds. Okay, because likelihood is generally measured in logarithmic terms. Okay, likelihood is generally measured in logarithmic terms, so you call it as log of odds or likelihood of odds, right? So, as yesterday I think you have been told that the LOD of 3 is used. Okay? The LOD of 3 means the likelihood of having the association is 100 times more or 1000 times more than that of not having the association. Okay? It is the likelihood of or likeliness of identifying the association is 1000 times more than that of not finding an association. That is what the LOD of 3 means, right? So, once you identify the LODs, you will be able to tell that whether this particular loci or locus is associated with the trait or not. Okay? That is what the basic thing is. Okay. Now, I will just skip on to this through this slides, because we have already explained this. This is how you make the populations, real population, backcross populations and DHLs. And some more thing, one more thing I wanted to just tell you, in certain cases like a perennial species, you will not be able to do this exercise. Yesterday he was telling 7 years of mango flowering, 7 years to get F1 flowered, then F2 will take another 7 years to, then somebody say he was, Dr. Akshay was telling he will retire, right? There are other methods. If you have natural populations, whose progenies are known, there are methods to identify or map or make populations which are looking genetically similar. They call it pseudo because it is not exactly the same. There are pseudo test crosses, there are pseudo back crosses, and you have half-zip progenies. All this can be used for mapping. Okay? I am not going much into detail about this. This is how what you do is you identify the difference between the population, the parent 1 and parent 2, identify the markers specific to the female parent, identify the markers specific to the male parent. If they are polymorphic, how they are distributed in the progenies, whatever progenies, whether it is test cross progeny or back cross progeny or the half-zip population, then you will be able to construct a map. You will be able to construct a map between these parents. Okay? You can have a map for the female parent, you can have a map for the male parent, then you can have the consensus map. Okay? That is possible. Okay? Now, as I said, we have talked about the markers. The marker can be random or linked. Random means they don't map to any trait. Linked means they are mapping, they are mapped to some traits, or they are associated or co-segregating with some traits. Okay? You may have markers on, they can have direct markers, you can have markers with LD, and you can have markers on LE, right? Okay? Then you will be having markers which are monomorphic and polymorphic, dominant and codominant, we have already discussed. Then you can have major genes and minor genes. Okay? Our interest is the QTL mapping is for minor genes, not for major genes. For major genes, you can use a method called bulk segregant analysis. Okay? If the gene is having a very significant influence on the trait, it can be a QTL also, a major QTL. We call it as a major QTL, which makes a significant difference between the parents. Then we call them, we can use bulk segregant analysis for doing this. And where we use 10 progenies bulk, from the extremes, then you map. Okay? And why the 10 is used, it is here. This is the formula to identify the probability of getting a band present or absent in a bulk of 10, right? 10 each. Okay? Now, coming to QTLs, the minor genes, this is the basic idea of mapping. As I said, make the cross, get the progenies, then genotype them, phenotype them, and then you do the analysis. Okay? What are all the analysis we can do? Okay, before that I will tell you what are the different types of QTLs. We can have QTLs that are additive, we can have QTLs that are dominant, or we can have QTLs that are epistatic or interacting, right? It is very easy to map the QTLs which are additive, then dominant, and it's very difficult to map QTLs that are epistatic. Okay? Now, as I said, this is the paradox. We know the genotype of the markers, and we don't know the genotype of the phenotype, right, or the trait. Okay? Then you have to combine them. Okay? These are the assumptions. As I said already, the genotypic values depend upon the genotype at the QTL locus or to the marker. This is exactly we are describing the paradox. Okay, leave this, I will go ahead. Now, marker QTL can segregate. Now, what is the idea is to identify a marker which is placed close to the gene, okay, or the QTL, right? Then now what are the options of segregations? When you have a marker M1 and an M2 and two QTLs Q1 and Q2, they can segregate independently, or they can overlap, they can still overlap, or they can overlap completely, they can overlap halfway. These are the different ways, these are different possible ways the QTL and the marker can segregate, right? Now let us see what are they. When there is no variation, we call it as there is no significant difference. If they vary between these two classes, these three classes, then we call it as slight variation, VQTL. If they are exactly your AA is in between these two, we call them as additive QTL. If one of them is closer to the one parent, we call it as dominant QTL. And if the A is better than the parents, then we call it as heterotic QTLs. Okay? You can call them in different ways. Right? Based on the expression pattern, the QTL can be classified into different ways. Right? Now, we have discussed this. The parents are polymorphic to be in nature, and they should have a variation between the traits, and they should be in linkage disequilibrium. And now, what are the possibilities of identifying the QTL between two markers, right? For example, you take, there are two QTLs, one QTL and two markers, right? In first case, you see that the QTL is in between the marker 1 and marker 2, right? Then you will be able to detect the QTL definitely, right? You will be able to detect the QTL. Now, if you take the second case, you have the QTL in between the markers, right? Okay, in first case there is the M2 is monomorphic. M2 is monomorphic. So in this case, the identification of the QTL will come only with the marker M1, right, marker M1. In second case, the M1 is monomorphic, M2 is monomorphic, but the QTL is there where you will not be able to map the QTL at all, right, you will not be able to map the QTL. In third case, you have the QTL which is monomorphic, so there is no question of mapping the QTL there, right, in this case. Fourth case, you have an additional marker which will help you to map the QTL, right, okay. Now, we have yesterday you have just gone through this, what are all the different methods. Yes, you have single marker analysis and the interval mapping. Then in single marker analysis, we use two methods called, one is called single factor analysis of variance or one way ANOVA, or otherwise called simple linear regression approach, okay. Both are one and the same in practical sense, which is only two different types of analysis of the same data, and we can call it as they are two sides of a same coin, okay. You are looking at the data in two different ways. The results are going to be exactly similar, and in here we said that this is a single marker analysis, that means one marker at a time is getting analyzed, okay. What we do here is, as I said, when you have two markers, when you have one marker having two alleles, one allele coming from parent 1, the other allele coming from parent 2, the progenies are classified based on parent 1 type and parent 2 type, right, based on the marker pattern. Now you have the trait values, phenotypic values for the particular group of genotypes, parent 1 types and parent 2 types. If the marker is associated with the trait, these two groups will have distinct variation in the trait. Is it not? If the marker is associated with the trait, for example, you have an allele A and a small a, and you have 10 plants here, 10 plants here. If the average of these 10 plants shows 50 centimeter height, and average of these 10 plants shows 100 centimeter height, then we say that the group 1 is different from group 2, right. How will you say that group 1 is different from group 2? Then you use statistics, okay. You use a single factor analysis of variance, where you say that the group 1 is statistically significant from the group 2 by doing the variance analysis, and if your variance ratio is higher than the expected value, then you say that they are distinctly different, statistically different. Once that is done, you say that the marker is associated with the trait. Two things you can calculate here: the percentage of variation explained, which will be equal to the between group sum of squares divided by total sum of square into 100 if you wanted to express that in percentage, okay. The ANOVA will give you three sources. One is between group S's, within group S's, and the error residual. What you have to do is find out the between group sum of squares, divide it by the total sum of squares, and multiply it with 100. You will get the percentage of variation explained, otherwise known as R square value, right. Second one is estimation of additive effect, where you have take the average of the parent 1, take the average of the parent 2 group, subtract them and divide by 2 or multiply with half, you will get the additive value, right, or that means the contribution of a particular allele towards the trait, okay. If the additive value is in minus, or if it is positive, you say that the parent 1 is the contributor of the trait, okay, or it is the contributor of the positive allele, okay, it is a contributor of the positive allele. In case if the additive value is negative, you call the contributor of the positive allele is the second parent, okay, because P1 is, we are subtracting P2 from P1. So, if the P2 is higher, you will have the negative value, okay. But as I said, as yesterday you learned that the single marker analysis is a rough estimate, and it can, many of the cases it can be overestimated. We will come to that. And next method is the interval mapping. We know that when you have two markers, 2 is better than 1, right? 2 is always better than 1. And if they are flanking, fantastic. So, that is the basic principle of interval mapping.
What about the peak marker? If we have a peak marker, isn't it better than if we use 2 markers?
If you,
on the other sides?
yeah, if you can get the peak marker straight a bit, but we actually we don't know at this point of time in the mapping, we don't have any idea about peak marker. If you have the peak marker with you, fantastic. But these are all random markers now we are using in the mapping process. So, the peak marker comes in the next point. Once having identified the QTLs, then you go closer to that interval, then you go into the fine mapping part, then you will be able to identify, pinpoint the peak marker. In random markers, sometimes we may be missing the peak marker, or sometimes we may have the peak marker. We don't know. It is not, we are not sure about. Whatever be the case, if peak marker is there, you are definitely going to get high R square value. If peak marker is not there, you will get a lesser R square value. Then you will have to converge it into to the peak marker. Okay. So, interval mapping uses the same principle, identify the two flanking markers between the QTL, then the next step is you try to estimate the likelihood of finding out the QTLs between these markers on a specific distance. For example, if these two markers are say 10 centimorgan apart, by maximum likelihood method, you will be able to estimate the values of the probabilities of the maximum likelihood for every 2 centimorgan distance. Once you have the probability of identifying the QTL at every centimorgan distance, you automatically can calculate the log value, right? Because it is a ratio. You can calculate the log value, then if the log value increases in between, you will be able to draw a graph. Then you get the peak graph, okay? Again, at this point of time, you don't know where the peak lies, because it is in between two markers which we don't know what are all the in between markers, right? Then to identify the marker for that peak, you will have to go for fine mapping. Right? So, there are two methods here, simple interval mapping, which is just as I said, walking in between the distance and estimating the thing. And the second one is the composite interval mapping, where we not only walk, we will also estimate, we use a model here. It is more statistically complex. We use a regression analysis, a backward and a forward and backward regression, stepwise regression analysis. Then you identify the walk distances, calculate the probabilities. Also you plant covariates, like you place some markers as covariates in the analysis. I am not going to the analysis model part now. You understand that you use some covariates in the model, so that you suppress the estimation of or overestimation of the marker probability. Okay? You suppress the overestimation of marker probability, use covariates. That is why the precision of the composite interval mapping is very high. Right? Okay, this we have discussed. The pros and cons of SIM it is better than the single marker analysis, but it is not as better as composite interval mapping. Okay. Then composite interval mapping, as I said, you have regression analysis and then a maximum likelihood, and this is a model estimate. Now comparing the output of simple interval mapping and composite interval mapping, you can see that composite interval mapping even precisely identify the QTLs that are close by, whereas in simple interval mapping that get overlapped. Okay, that get overlapped. Okay. Now further going into deep, you can have mixed model mapping or Bayesian model mappings, where you will be able to estimate the environmental interactions of the QTL by environment interactions, and also you will be able to estimate the epistatic interactions, right. These are all more complicated way to analyze the QTLs. Now this is the phenotypic influence of a minor effect QTL. I am not going much into this. These are simple things. When the environment complicates the QTLs, sometimes when two QTLs are there, the environmental effects is more, then there seems to be a one QTL. But if the environmental effect is removed, you can identify them as two different QTLs. Okay. Then most important thing is in the QTL mapping is you generally we land with a very good genotypic data, but we land with a very poor phenotypic data. So the phenotypic data must be very precise to have a QTL mapping. For that you must use valid statistical designs and proper care of the materials to get the phenotypic data. Once that is done, your QTL mapping is generally used to be fine. And having identified a QTL, you have to validate it. Yesterday it was being emphasized. Validation is very important. You can have it on multiple years, multiple locations, multiple populations. Validation is very important. And everywhere the threat is there, the overestimation or underestimation, and which is otherwise called the Bevis effect, which depends on mating systems, number of recombinations, hotspots, epistasis, method of analysis, environmental influence, human error, number of QTLs, and so on. Okay. The Bevis effect is very important to be taken care of, because when as in this picture shows, the number of populations are less, you tend to overestimate. When the populations increases, Bevis actually found out, when he said that in his simulation study, he found out that if you use 100 individuals, the QTLs are overestimated. If you use 500, it is slightly better, and if there are 1000 individuals, the QTLs are more realistic. That is what the Bevis effect is in his paper. That is what he has described. And making me then having identified the QTL, then what we have to do is we have to go for further fine mapping, identifying the gene, then use them for map based cloning or whatever, then go for marker assisted selection and so on, right. Okay. Then there are several methods to use, several tools for getting the QTL analysis done, some of which we will see in the afternoon. And finally, what I have to tell is that you have to keep in mind that the QTL is not a gene. Once you identify the QTL, I understand that they are not the gene, they are the region where you are likely to identify the gene. Okay? So never confuse that QTL is a gene. Thank you.
Thank you, sir. Thank you very much for eloquent presentation. It was a glimpse of quantitative genetics combined with molecular markers. And he has given a very clear picture on how QTL mapping is done and how the softwares also work. I'm sure he will be having more of stuff to